In this video, we will be discussing troubleshooting bacterial transformations. So what are bacterial transformations? Bacterial transformations is a process of horizontal gene transfer where bacteria take up foreign material from the environment. It acts to increase genetic variability amongst bacteria. This technique has been used in numerous research endeavors, such as Avery et al. Determining that DNA is a hereditary material in 1944. During a bacterial transformation, competent cells are exposed to recombinant DNA. The permeabilized membrane allows the DNA to pass through and integrate itself into the host genome, or remain as a plasmid. There are several different methods to permeabilize the membrane, such as chemical transformations, which utilize heat shocking to create the pores within the membrane, or electroporation, which utilizes electric shocks to create the permeable membrane. Following cell recovery after the transformation, ideal plates will have an acceptable range of colonies present. As well, upon DNA sequencing technologies such as gel electrophoresis, the transformed bacteria will also contain the whole plasmid insert. During bacterial transformations, there are various errors that can be encountered. You may find that your plate contains transformants with incorrect or truncated DNA inserts, or the transformants may have empty vectors where there are no DNA inserts. You may also observe numerous overgrown clumped colonies, or many colonies with no transforming DNA. Another error you may encounter are plates with few or no transformants. Each of these errors are caused by a variety of factors. For example, if you observe few or no transformants during your experiment, this can be caused by a variety of reasons and factors. Some causes could be suboptimal transformation efficiency, which could be tested through adding a positive control. Another cause could be suboptimal time and temperature, which can be overcome by ensuring that the incubator is set to the proper temperature and there's a sufficient amount of time allowed for recovery and plating. Another cause could be that an insufficient number of cells were plated during the transformation, which can be overcome by recovering the cells within the appropriate medium and allowing ample time for growth. Despite the various errors that you can encounter during the transformation process, there's a simple yet effective troubleshooting guide, allowing you to determine where the error is made. We will take a closer look at the following components of the process: the protocol, cells, plasmid DNA, and the antibiotics used. When an error is initially encountered, it is important to review the protocol again, as mistakes do occur. Some labs may utilize an outdated protocol, which could affect the experiment's results. It's also important to follow the protocol, particularly the timing and temperature of some procedures. While timing and temperature do not have to be strictly adhered to at all times, such as during incubation, it must be followed during vital aspects of the procedure, such as heat shocking. Prolonged exposure to heat or exposure to higher temperatures can have a drastic impact on the experiment as the plasmids can denature. Prolonged exposure to heat or high temperatures can cause the bonds within the plasmid molecule to break down, inhibiting the transformation process. Timing between transformations and plating also impact the transformation efficiency. Depending on the strain in plasmid, an increase in the interval between transformation and plating decreases the transformation efficiency significantly. After analyzing the protocol and ensuring the steps are adhered, the next component to review are the competent cells. Since some strains are not naturally competent to take up foreign DNA, they must be pretreated to ensure that DNA is unable to pass through the membrane. To produce these competent cells, heat shock, chemicals, electric impulses, or a combination of these three methods can be used. Often, cells in the logarithmic growth phase are treated for transformations. If no colonies were observed, the viability of the competent cells can be quickly determined through the inoculation of an LB plate. If bacterial colonies form, then the cells are viable. During this phase, you can also transform the cells with an uncut plasmid to determine the transformation efficiency, which is the number of transformants generated per microgram of supercoiled DNA used in the transformation reaction. An ideal transformation efficiency is greater than 10 to the power 4. Another aspect to inspect is the DNA plasmid being utilized. For example, the size of the plasmid impacts the transformation efficiency of the plasmid. Large plasmids have a lower transformation efficiency than small plasmids, indicating to researchers that a different electroporation method may have to be utilized. As well, it is important to ensure that an adequate amount of plasmid data has been added Ideally, you want to use between 10 to 100 nanograms of DNA in total. The final aspect to inspect are the antibiotics used during the transformations, as antibiotic resistance is a common selection marker for bacterial transformation. It is vital to ensure that the correct marker is added. Transformed bacteria are selected for their ability to multiply and form colonies in the presence of the antibiotic. Expression of an antibiotic resistant gene present on the plasmid allows only the transformed bacteria to degrade the antibiotic, thus allowing them to propagate while untransformed cells die off. To control for this, simply isolate one of your cells with your confidence cells. If they do not grow with the plates, then they have the correct antibiotic. To conclude, bacterial transformation play a crucial role in many research endeavors. Although it is a simple process, there are many possibilities for errors to occur. To troubleshoot negative or inconclusive results, examining the protocol, the cells, the plasmid, DNA, and the antibiotic used during the experiment allows the researchers to pinpoint where their experiment went wrong. Thank you for watching the video, and we hope this helps you in your future lab endeavors. Resources used for the presentation will be credited in the description box below.